Meselson and Stahl experiment. Matthew Meselson and Franklin Stahl performed the experiment in 1958, which supported the hypothesis that DNA replication takes place by semi-conservative method of replication. In this method, new DNA molecule which is formed after replication contain one old strand and one new strand. In this experiment, they used bacteria E. coli with the technique of density gradient centrifugation, which separate the molecule on the basis of their density. So there are total 4 steps in this experiment. First is the calibration step. In this experiment, they took the E. coli bacteria and cultured it in the ammonium chloride containing the isotope N14 of nitrogen. As we know that DNA is made up of 2 strands, that is 2 polynucleotide chain, and the E. coli is developed in the medium containing the N14 isotope. Therefore, both the nucleotide chain contain the N14 isotope of nitrogen, and after the density gradient centrifugation technique, they found the light DNA band. In the second step is also calibration step. In this, they use the E. coli bacteria and culture it in the medium containing the ammonium chloride, but there is a change in the isotope. It contains the isotope N15. As the culture medium contain the N15 isotope, both the nucleotide chain contain the N15, and after the density gradient centrifugation, they found the heavy DNA band. Now, in the third step, they use the heavy bacterium containing the N15 polynucleotide chain, and they culture this bacteria in the ammonium chloride, which contain the isotope N14. Okay, after the replication, the 2 chains which are produced are neither heavy nor light. As a result, after the density gradient centrifugation technique, they found the intermediate DNA strand. Okay, so last is the fourth step, in which they use the hybrid bacteria, that is N15, N14, and that hybrid bacterium is grown in the medium containing the ammonium chloride having the isotope N14. So after the centrifugation, they found the 2 bands of DNA, that is, one is a light DNA band, another one is an intermediate band. So these are the 4 steps performed during this experiment. Now let's see what conclusion they put forth after this 4 step. So in the first calibration step, they found light DNA band. In the second calibration step, they found the heavy DNA band. In the third step, they found intermediate DNA, while in the fourth, they found the light DNA and intermediate DNA. So this step 3 eliminate the conservative replication mechanism, as they found the intermediate DNA band, because we know that in the conservative replication, one of the DNA is contain the parental strand, both the parental strand. As we know that E. coli bacteria which we use in the third step is a heavy bacterium, that is N15. So there must be have the heavy DNA band, but as it is only intermediate DNA band, so this eliminate the conservative replication mechanism, while step 4 eliminate the dispersive replication mechanism. Okay, by conclusion from the step 3 and the step 4, they declare that the DNA takes place or synthesized by the semi-conservative method of replication due to the presence of intermediate strand in the both step. Thank you.